Scientists just tested a vaccine designed for viruses that may not exist in humans yet. Not a prediction app, not a sci-fi cure, a real phase 1 human trial built around a strange idea. Instead of chasing the latest variant after it shows up, design a vaccine from the pieces a whole virus family has in common. That is a promise behind a candidate vaccine called pEVAC-PS. Cambridge described it as using an AI-designed super-antigen. PubMed calls it a first-in-human, needle-free dose-escalation study of a candidate pan-Sarbeco virus vaccine. And that difference matters, because the headline sounds like AI built a vaccine for the next pandemic. The reality is more interesting. Scientists are trying to make vaccines less reactive by training the immune system on the family resemblance of dangerous viruses. So the real question is not, did AI solve pandemics? The real question is, can a vaccine be built before we even know which virus is coming? On June 5, 2026, the University of Cambridge announced results from the first human clinical trial of a universal Sarbeco coronavirus vaccine. Sarbeco viruses are not one virus, they are a group. SARS coronavirus 2 is in that group. So is the original SARS virus. So are related animal viruses, including bat viruses, that could potentially spill over into humans someday. That is the target. Not every virus, not every pandemic, not a magic shield around the planet. This candidate vaccine is aimed at a particular branch of the coronavirus family tree. The trial included 39 healthy volunteers ages 18 to 50 who had already received 2 or 3 COVID-19 vaccine doses. It used needle-free delivery through the skin. The main phase 1 question was safety. The reported result? Well tolerated with no significant safety concerns identified. That is important, but it's not the same as proving protection. Phase 1 tells you whether the candidate looks safe enough to keep studying and whether the immune system responds in a way worth following. The viral version of this story wants a victory lap. The scientific version is still at the starting line. Viruses change. Some change slowly. Some change fast enough that public health has to keep updating the target. That's why the phrase variants became normal during COVID. It's also why flu vaccines are updated regularly. Scientists watch what's circulating, make a best call, then manufacturers produce vaccines against strains that already exist. That system can work, but it's reactive. Surveillance, manufacturing, and distribution take time. A virus doesn't wait politely while the paperwork finishes. The Cambridge team is trying a different approach. Instead of asking what exact variant is circulating right now, they ask what parts of this whole virus group keep showing up again and again. The goal is not to outguess one future virus. The goal is to find the shared machinery that many related viruses can't easily throw away. A vaccine needs an antigen, a piece or design that teaches the immune system what to notice. Traditional vaccines often use antigens from a specific strain or variant that can work when the future looks like the present. But when the virus mutates enough, the match gets weaker. The Cambridge article says this team used genetic sequence data from Sarbeco viruses logged by surveillance programs around the world. Then, using machine learning, they designed a super-antigen containing features common across the group, including viruses that haven't emerged in humans yet. That's the AI part, not crystal ball, pattern finder. The computer is not saying, here's the next pandemic on Tuesday. It's helping researchers search a massive library of viral sequences and ask which parts are shared, important, and visible enough to make a useful vaccine target. The trial name is less flashy, pEVAC-PS. The platform is dose-escalation, short for digitally immune optimized synthetic vaccines. And the paper's abstract says the candidate was selected preclinically for its ability to induce broadly protective immune responses across Sarbeco viruses, including SARS, SARS coronavirus 2, and related viruses representing potential zoonotic spillovers. That phrase, zoonotic spillover, means a pathogen jumping from animals into humans. WHO's research and development blueprint even includes Disease X, not as a secret named virus, but as a placeholder for a serious epidemic caused by a pathogen not currently known to cause human disease. So the vaccine isn't just about the last pandemic. It's about building tools for the unknown one. The pEVAC-PS trial was open-label, phase 1, and dose-escalating. Volunteers received 2 doses, one at day 0 and one at day 28. Researchers tested different dose levels delivered into the skin using a PharmaJet Tropis needle-free device. That could matter if a vaccine ever needs fast deployment in places where conventional injections are harder to run. The primary outcomes were safety and reactogenicity. Did it cause concerning side effects, and how did people react? The PubMed abstract says the vaccine was safe and well tolerated. Then comes the immune response. The abstract says immunogenicity was modest, partly because the trial was complicated by real life. Participants already had high baseline antibody levels, prior COVID vaccinations, and uneven Omicron exposure histories. That makes the immune results harder to interpret. Still, participants developed measurable responses to conserved Sarbeco virus epitopes. That supports the feasibility of the design strategy. That is the sober center of the story, not pandemic solved, feasibility supported. If this approach works, the bigger story is not one shot, it is a new way to think about vaccine design. Instead of waiting until a virus is already spreading in humans, researchers could build candidate vaccines around dangerous virus families, coronaviruses, influenza, Ebola group viruses, and other pathogens with pandemic potential. That does not remove uncertainty, it moves the work earlier. That is the real future-proofing idea, not guaranteed protection forever, but more preparedness before the emergency. Some designs will fail, but if even a few work, the next outbreak response could start from a shelf of research tools instead of a blank page. That's why Disease X matters. It's not a doomsday slogan, it's a planning category. Prepare platforms, targets, trials, manufacturing plans, and safety data before panic starts. There are 2 easy mistakes with this story. The first is hype. AI built a universal vaccine so pandemics are over. No. This was a small phase 1 trial. It did not prove real-world protection. It did not show that vaccinated people can walk through a future outbreak unharmed. It did not test every coronavirus, let alone every virus. The second is dismissal. If it's not proven protection yet, then it means nothing. Also no. Early trials are how medical tools become real or fail honestly. Safety matters. Measurable immune responses matter. A design strategy that can be tested in humans matters. The interesting part lives between the extremes. Less reaction, more anticipation. The next step is larger phase 2 testing. Cambridge says that will assess immune responses in a wider and more diverse population. That's where harder questions show up. Will responses be strong enough? Will they last? Will they protect against infection, severe disease, or both? Will older adults and people with different immune histories respond the same way? And the biggest question, if a future animal virus spills over, will the shared features chosen by the algorithm still be the right ones? That's the tension. The vaccine is designed for breadth, but nature is not obligated to cooperate. Viruses mutate, immune systems vary, trials are expensive, manufacturing is political, distribution is unequal. Public trust can make or break even a technically good vaccine. What makes this story feel like science fiction isn't the word AI, it's the scale of the library. Every viral sequence collected by surveillance programs is a small clue. A bat virus in one region, a human outbreak in another, a mutation that appears, disappears, or keeps coming back. Together, those clues become a map of what viruses share. That map is what machine learning can help search. The future vaccine may not come from predicting one perfect enemy. It may come from noticing the family resemblance among many possible enemies, then choosing a target that stays stable enough to matter. The next pandemic might begin with a spillover event, but pandemic preparation begins much earlier in sequence databases, animal surveillance, trial design, manufacturing plans, and the unglamorous work of proving a clever idea is safe in real humans. The most important word in this story isn't AI, it's before. Before the virus is everywhere, before hospitals fill, before vaccine makers are forced to sprint after a moving target. pEVAC-PS is not proof that we can stop the next pandemic, but it is proof that researchers are trying to move the battlefield from reaction to preparation, from one variant to a family tree, from a known enemy to the shape of possible enemies. And that's why this small trial matters, because one day, the most important vaccine may not be the one designed after the outbreak starts. It may be the one that was waiting before anyone knew its name. Reality isn't boring. Reality has lore.